This is the first study to use the wisdom of crowds to optimize the use of omics data in maternal blood to predict gestational age and the risk of preterm birth. The objectives of this study were to assess the value of molecular markers in maternal blood to predict gestational age and spontaneous preterm birth. The study was performed by engaging the scientific community in a collaborative competition under the DREAM Challenges Project, which stands for Dialogue on Reverse Engineering Assessment and Methods. This approach has contributed meaningful solutions in other areas of medicine and biology. The DREAM Challenge framework explores multiple methods proposed by participants in the challenge and also provides a robust assessment of the analytical methods and results. The first aim, to predict gestational age based on maternal blood biomarkers, will be presented next. Currently, the standard method to assess gestational age is based on the date of the last menstrual period and fetal ultrasound biometry. Alternative approaches are desirable. Our first objective was to determine whether maternal blood RNAs can predict gestational age. We performed a retrospective longitudinal case control study that included women with normal pregnancies and those who subsequently developed common complications of pregnancy. Women were enrolled and followed until delivery at the prenatal clinic of the Perinatology Research Branch of NICHD National Institutes of Health in Detroit, Michigan. Maternal blood was analyzed to characterize the transcriptome in each sample using Affymetrix human transcriptome arrays. The study design is shown in this figure. Each line represents one patient. The circles correspond to the time of sample collection, and the color identifies the clinical status, normal or complicated pregnancy, adjudicated after delivery. Samples were divided into a training set for model development and then a test set to assess the performance of such model. Participants in this DREAM challenge were asked to build a prediction model of gestational age based on the training samples and subsequently to apply the model to data in the test set of samples and to submit their results via an online portal. The accuracy of predictions on the test set were then used to rank the different analytical approaches. We found that blood RNA profiles significantly predicted gestational age, as defined by ultrasound, in patients who had a normal pregnancy, as well as in those with different pregnancy complications, including preeclampsia, preterm prelabor rupture of membranes, and preterm labor and delivery with intact membranes. The genes that contributed the most to the prediction of gestational age were those involved in the regulation of the immune system, inflammation, and developmental processes. This supports the concept of an immune clock of pregnancy. Therefore, these RNAs could serve as candidate biomarkers to accurately date gestations based on maternal blood samples. The second objective was predicting the patient at risk for spontaneous preterm birth. This is important because preterm birth affects 15 million neonates each year, and 2/3 are due to spontaneous preterm labor with either ruptured or intact membranes. Preterm labor is not a single condition, but is a syndrome caused by multiple mechanisms of disease. For the prediction of spontaneous preterm labor, we use transcriptomics, as well as maternal plasma proteomics data. This approach was used to evaluate prediction within the study cohort of the Perinatology Research Branch of NICHD NIH in Detroit, and within a separate cohort contributed from Canadian investigators. Importantly, cross-cohort evaluation was also performed. All patients were asymptomatic at the time of sample collection. Participants in the DREAM Challenge submitted computer programs to select the optimal timing of measurements and the combination of biomarkers to calculate the risk score for preterm birth. One of the measures of prediction performance was the area under the receiver operating characteristic, or ROC curve, which shows sensitivity of a model in the vertical axis and the false positive rate in the horizontal axis. When the sensitivity is the same as the false positive rate, the area under the curve is 50%, and the prediction model is no better than a random guess. In contrast, when the model reaches 100% sensitivity with a 0% false positive rate, the area under the curve reaches the ideal value of 1. The prediction algorithms proposed by the participants in the DREAM Challenge were ranked by the area under the curve and other performance metrics under multiple scenarios defined by omics platforms, cohorts, and gestational age at which the sample was obtained. For example, maternal blood RNAs at 27 to 33 weeks predicted preterm pre-labor rupture of membranes. When expression data at an earlier gestational age was included in the model for the same genes selected at 27 to 33 weeks, cross-cohort predictions improved. This indicates that there is consistency in gene expression dysregulation across time points and cohorts. Genes predictive of subsequent preterm pre-labor rupture of membranes were related to leukocyte-mediated immunity, among other pathways. Blood proteomic profiles also predicted spontaneous preterm delivery with intact membranes at 17 to 22 weeks and also 27 to 33 weeks. Plasma proteomics was more informative than cellular transcriptomics, and the combination of the two platforms appears to increase accuracy. The perturbation in maternal plasma proteome was detectable prior to spontaneous preterm birth, and protein dysregulation was consistent across time points for women with preterm labor and intact membranes. There were also common proteins dysregulated between cases of preterm labor with intact membranes and cases with preterm prom at 27 to 33 weeks. These proteomic markers included known mediators of prematurity, such as interleukin-6, but also new candidate proteins. These results were possible by harnessing what is called the wisdom of crowds to optimize the use of omics data to address research questions in pregnancy. The DREAM Preterm Birth Prediction Challenge was based on data generated at the Perinatology Research Branch of NICHD, NIH, and Wayne State University, and elsewhere. The financial incentive to participants in the challenge was funding their attendance to the DREAM Challenge Symposium. Such incentives were provided by Wayne State University School of Medicine and the March of Dimes. The top-ranked teams of the first challenge focused on the prediction of gestational age were Dr. Pataki and Dr. Sabai from Budapest, Hungary, and Dr. Guan from the University of Michigan. For the second challenge focused on prediction of preterm birth, the top-ranked teams were Dr. Pataki and Dr. Sabai from Budapest, Hungary, and Dr. Guan from the University of Michigan, and Dr. Kutum from New Delhi, India. The conclusions of this study were maternal blood RNAs predict gestational age in normal and complicated pregnancies. Changes in maternal blood transcriptomics predict spontaneous preterm prom, and repeated measurements increase accuracy. And plasma proteomic profiles predict spontaneous preterm labor and birth. This study was published in the journal Cell Reports Medicine.